eukaryotic cell. And here we will first start with a group of cell organelles under a common heading. The endomembrane system. Actually, in the cytoplasm of eukaryotes, there are two types of cell organelles. One, those cell organelles whose functions are coordinated. Mane, those cell organelles in the cytoplasm of eukaryotes which work as a group. And number two, those cell organelles whose functions are independent of each other. The first group of cell organelles, I will write it here. The group of cell organelles whose functions are coordinated. Mane, group of cell organelles which work in coordination with each other, which work in harmony with each other, whose functions are dependent upon each other. All those cell organelles whose functions are dependent upon each other or whose functions are coordinated in the cytoplasm of eukaryotes, we together call them as endomembrane system. I hope everyone is listening. All the cell organelles which are included in this endomembrane system have had one thing common, and that is all of them are single membrane bound structures. जितने भी सेल ऑर्गेनेल्स को आप लोग एंडोमेम्ब्रेन सिस्टम में इंक्लूड करेंगे, उन सब में एक कॉमन बात क्या है? All of them have a single membrane around. So let us see which cellular organelles we need to include in this endomembrane system. Number one, we have the famous endoplasmic reticulum. Endoplasmic reticulum. Number two, we have the Golgi apparatus. नाम सुने ही होंगे आपने. Number three, we have the lysosomes. And number four, the vacuole. This is a set of four cell organelles, and this set of four cell organelles together constitutes the endomembrane system of eukaryotic cells. Mane, ER, Golgi apparatus, lysosomes, and vacuoles. They work in coordination with each other. And all the four have had one thing common, that is, they are single membrane bound cell organelles. Harsharan Kaur is listening. Faizan is listening. I hope you people are. So let us one by one discuss these four members of this endomembrane system. Let us get started with the endoplasmic reticulum. Endoplasmic reticulum आप लोग heading देंगे. This endoplasmic reticulum, you all may be knowing, is abbreviated as ER. फिलहाल, let us leave the bookish information. अपना guess करते हैं पहले. रेटिकुलम वाला टर्म जो है, ये बायोलॉजी में यूज होता है नेटवर्क लाइक स्ट्रक्चर्स के लिए. Reticulum in biology means network-like structures. And if these network-like structures are present in a to plasma membrane. If the network-like structures are present inner to plasma membrane, endo means inner, plasmic means plasma membrane, then we use the term endoplasmic reticulum. Mane, we have some network-like structures which are present inner to the plasma membrane of eukaryotic cells. Inner to the plasma membrane of eukaryotic cells, mane into the cytoplasm. The credit for the observation of these endoplasmic reticular structures goes to Garnier. Mane, Garnier first of all observed these network-like structures inner to the plasma membrane of eukaryotic cells, that is in the cytoplasm. Garnier, for these network-like structures, used the term ergastoplasm रेटिक्यूल. गार्नियर ने इन नेटवर्क लाइक स्ट्रक्चर्स के लिए विच ही ऑब्जर्व इन द साइटोप्लाज्म क्या टर्म यूज किया? ergastoplasmic reticulum. Now, reticulum, you know, network-like structures. As per Garnier, these reticulum-like structures or network-like structures are present where? In the ergastoplasm. गार्नियर ने ये नेटवर्क लाइक स्ट्रक्चर्स कहां देखे? सर, ergastoplasm में देखे, इसीलिए नाम रखा ergastoplasm. Ergastoplasm is basically a non-living part of cytoplasm. Ergastoplasm is basically what? Sir, it is a non-living part of cytoplasm. Let us make this thing more easy for you people by supposing. To give you an actual idea of ergastoplasm, let us suppose we have a cell over here. Suppose this yellow color gola is a cell. Cell will be having a well-defined plasma membrane, and inside this plasma membrane, the green color dots are going to represent cytoplasm. Let me label these green color dots as cytoplasm. Cytoplasm of eukaryotic cells is divided into two parts. यूकैरियोटिक सेल्स का जो साइटोप्लाज्म होता है उसको आप डिवाइड करेंगे दो पार्ट्स. सपोज ये एक पार्ट है और ये दूसरा. इन वन पार्ट ऑफ दिस cell cytoplasm we have nucleus, we have other cell organelles. माने ये वो वाला पार्ट है साइटोप्लाज्म का where cell organelles are present. और ये वो वाला पार्ट है साइटोप्लाज्म का जो मैं आपको पिक्चर पे दिखा रहा हूं. ये वो वाला पार्ट है साइटोप्लाज्म का where reserve food materials or waste products are present. बिल्कुल आराम से सुनिएगा, हमारा इशू क्या है सर? आवर इशू इज व्हाई गार्नियर यूज्ड द टर्म ergastoplasmic reticulum for ER. Reason? He observed them in the ergastoplasm of cytoplasm. Ergastoplasm is what? Sorry, it is a non-living part of cytoplasm. And in that context, what you know up to now, sir, we know the cytoplasm of eukaryotic cells is divided into two compartments. One part of cytoplasm where cell organelles are present, and second part of cytoplasm where reserve food materials or waste products are present. Is that clear? Now, this part of cytoplasm where cell organelles are present, obviously here metabolic activities will occur. This part where cell organelles are present, here metabolic activities will be taking place. All of us know metabolic activities are the living reactions. So this part of cytoplasm where cell organelles are present and hence metabolic activities occur constitutes the living part. This living part of cytoplasm we call as protoplasm. इसको क्या कहते हैं? Protoplasm. On the other hand, the part of cytoplasm where reserve food materials are seen, where waste products are seen, here no metabolism or no metabolic activities. Why there are no metabolic activities? Sir, because there are no cell organelles. This part of cytoplasm where there are no cell organelles, hence no cellular activities, constitutes the non-living part of cytoplasm. And this non-living part we call as ergastoplasm, or it is sometimes also called as deutoplasm. A quick recap. Garnier first of all observed ER as network-like structures in the ergastoplasm. Hence he named them as ergastoplasmic reticulum. ergastoplasm में आपको क्या समझ आया? सर हमें ये समझ आया कि it is non-living part of cytoplasm, otherwise known as deutoplasm. Here no cellular activity will take place, only reserve food materials and the waste products generated out of various reactions are seen. Here, first of all, Garnier observed these ER structures and hence called them as ergastoplasmic reticulum. Is that okay? Is that okay? Now, the actual credit for the discovery of ER goes to Porter and Thompson. 1945. 1945 में सबसे पहले पोर्टर और थॉम्पसन ने किनको किया था एक्चुअल में डिस्कवर? ER को. And for the first time, Porter used the term endoplasmic reticulum For the first time, Porter used the term endoplasmic reticulum or ER for a fine network of tiny tubular structures, tiny tubular structures which are present scattered in the cytoplasm. In the cytoplasm. तो एक्चुअल में एंडोप्लाज्मिक रेटिकुलम क्या है? इट इज अ नेटवर्क ऑफ tiny tubular structures which are present scattered in the cytoplasm. This definition of ER is asked as it is in the competition, that a network of tiny tubular structures which are present scattered in the cytoplasm are which of the following cell organelles? Answer: endoplasmic. So the actual credit for the discovery goes to Porter and Thomson. Porter used the term ER for a network of tiny tubular structures, small tube-like structures, which are present scattered in the cytoplasm. This is how endoplasmic reticulum was discovered. I hope every item is getting these things clear. Now, from competition point of view, in this year, we have three important focus areas. माने एंडोप्लाज्मिक रेटिकुलम से आपके एग्जाम में तीन एरिया से क्वेश्चंस पूछे जा सकते हैं. Although we have many more areas to explore in the year, but from NEET point of view, we will discuss only three important aspects of ER. One, the origin of ER. Second, the ultrastructure of ER. And third, the functions of ER. From these three areas, you can be asked a question in the examination. We have many other areas to explore in this ER, like its enzymatic machinery, like its genetics, like its mechanism of function, like its wear and tear. But from NEET point of view, we will focus only on these three areas. So let us first of all come towards the origin. Origin of whom? ER. That is endoplasmic reticulum.
Class.
Endoplasmic reticulum develops due to the evagination of nuclear membrane or nuclear envelope. We all know that all the biomembranes are elastic. So same would be true for nuclear envelope. जैसे plasma membrane is elastic, same is the case with nuclear membrane. It is also elastic like plasma membrane. So we are going to develop endoplasmic reticulum from the nuclear envelope. How come? Let me show you. Let us consider. Consider what? We have a cell over here. Let us draw a cell over here. Eukaryotic cell. Let us draw a eukaryotic cell over here. To show the origin of what? Endoplasmic reticulum. Suppose We have a cell over here. Sorry, cell cell. In this cell, beside cell wall, suppose this is the cell wall. We are going to have a well defined plasma membrane जिसके अंदर हमें दिखाना है ये नेटवर्क बोले तो एंडोप्लाज्मिक रेटिकुलम. This green color cover you can consider as the plasma membrane of this cell. Inside this plasma membrane, we need to show this tube like network structure, जिसको हम ER कहेंगे. इसको हम करेंगे tag as cytoplasm. The red color dots, let us suppose, represent the cytoplasm. In this cytoplasm, we are going to come across a well-defined nucleus. Suppose this is the outer membrane of nucleus, and this is the inner membrane. इसको हम करेंगे टैग एज न्यूक्लियस. मैंने न्यूक्लियस को दिखाया है आधा, क्योंकि मुझे उससे ईआर निकालना है. जैसा कि मैंने ऊपर लिखा, दिस ईआर डेवलप्स थ्रू द इन्वेजिनेशन ऑफ न्यूक्लियर. इस न्यूक्लियस का जो आउटर मेम्ब्रेन होगा, आउटर मेम्ब्रेन, किसका आउटर मेम्ब्रेन? सर न्यूक्लियस का. उसमें होगी इवेजिनेशन लाइक दिस, ऐसे 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 और ऐसे. किसमें होगी इवेजिनेशन? सर न्यूक्लियर मेम्ब्रेन के आउटर वाले हिस्से में इवेजिनेशन होगी, और उस इवेजिनेशन की वजह से डेवलप होगा व्हाट वी कॉल एज द एंडोप्लाज्मिक रेटिकुलम. समझ में आई आपको बात? जब भी आपको एंडोप्लाज्मिक रेटिकुलम के ओरिजिन की बात करनी हो, तो इट ऑलवेज अराइजेस फ्रॉम द आउटर मेम्ब्रेन ऑफ न्यूक्लियर एनवेलप. हाउ कम? थ्रू इट्स इवेजिनेशन. थ्रू इट्स व्हाट? इवेजिनेशन. दिस इज all regarding the origin of endoplasmic reticulum. Now, the second aspect regarding this ER which we need to explore is its ultrastructure. ER के ultrastructure की बात करते हैं. Here I am going to write it. Ultra structure of ER. Ultrastructure, I mean to say electron microscopic structure of endoplasmic reticulum. Electron microscopic details reveal that this ER is made of three structural components. There are three structural components of ER, or there are three structural elements of ER. Number one, we have cistern. The first structural element of ER, cisternae. Number two, we have tubules. And number three, we have vesicles. The three structural components of ER include cisternae, include tubules, include vesicles. Let me show you the visuals of cisternae first. मैं आपको cisternae के visuals दिखा. The things are going to be like this. Like this. ये मैं क्या दिखा रहा हूं सर? आप ER का पहला स्ट्रक्चरल कंपोनेंट दिखा रहे हैं, सिस्टर. The things are going to be like this. Like this. These are sister. और इन सिस्टर ने के ऊपर आप छोटे छोटे गोले बनाएंगे. Like this.
Yes, sir.
The first structural element of ER, sister. अगर आप लोगों को अपनी स्क्रीन पे सिस्टर ने की ड्राइंग दिख रही है तो नंबर वन आपको ये दिख रहा होगा, सिस्टर ने आर लॉन्ग, फ्लैट, ट्यूब लाइक, unbranched structures. नंबर टू आपको ये लग रहा होगा, these long, flat, tube-like, unbranched structures are staked over one another. Over one another. माने these cisternae are adjusted over one another, and this adjustment of cisternae over one another is called their staking. माने एग्जाम में क्वेश्चन आएगा, which structural components of ER are stacked? Answer, cisternae. Number three, these cisternae are continuous with the outer membrane of nuclear or nucleus. सिस्टरने के बारे में तीन इंपॉर्टेंट इंफॉर्मेशन. दूसरा वाला इंफॉर्मेशन सबसे ज्यादा इंपॉर्टेंट. पहला, दे आर लॉन्ग, फ्लैट, ट्यूब लाइक, अनब्रांचेड स्ट्रक्चर्स. येलो शेड्स आपको दिख रहे हैं. These long, flat tubes are unbranched and they are staked over one another. They are continuous with the outer membrane of nucleus. Sir, क्या मतलब these cisternae are continuous with the outer membrane of nucleus? माने यहाँ पे जो मैंने ये ब्लू कलर के ER structures दिखाए हैं, which are continuous with the outer membrane of nucleus, these are actually cisternae. When I say cisternae are continuous with the outer membrane of nucleus, तो ये ऊपर वाली सेल की ड्राइंग देख के आप ये आइडिया लगाएंगे कि सर ने जो न्यूक्लियर मेम्ब्रेन के साथ ER दिखाया है वो एक्चुअली सिस्टरने दिखाया है. Is that okay? Plus, these long, flat, unbranched tube-like structures are characterized by the presence of ribosomes on their outer surface. outer surface. सिस्टर ने की आउटर सरफेस पे आपको छोटे छोटे ग्रेन्युलर स्ट्रक्चर्स दिखेंगे. दोज ग्रेन्युलर स्ट्रक्चर्स आर राइबोसोम्स. कौन से राइबोसोम्स? एटीएस राइबोसोम्स. These ATS ribosomes, which are attached with the outer surface of cistern, are called cytoplasmic ribosomes. They are called cytoplasmic ribosomes. These ATS ribosomes, that is, which are attached with the outer surface of cistern, you may be knowing they might have a smaller subunit and a larger subunit. एटीएस राइबोसोम्स के दो सब यूनिट्स होते हैं, स्मॉलर सब यूनिट एंड लार्जर सब यूनिट. स्मॉलर सब यूनिट 40s एंड लार्जर सब यूनिट 60s. सर आप ऐसा क्यों कह रहे हैं? आप राइबोसोम क्यों पढ़ा रहे हैं हमें? मैं बिल्कुल आपको राइबोसोम नहीं पढ़ा रहा. मुझे सिर्फ ये दिखाना है हाउ दिस एटीएस राइबोसोम्स बाइंड विथ the outer surface of cistern. ATS ribosomes use their larger subunit in order to bind with cistern. माने with the help of their larger subunit, these ATS ribosomes, they attach themselves with the outer surface of cistern. माने ये जो छोटे छोटे आपको ग्रेन्यूल्स दिख रहे हैं येलो ट्यूब्स के ऊपर, ये एक्चुअली ATS राइबोसोम्स है जो अपने लार्जर सब यूनिट को जोड़ेंगे किसके साथ? सिस्टर्न के साथ. सर कैसे? वो ऐसे कि ये जो 60s सब यूनिट होगा, it uses two important glycoproteins. Two important glycoproteins are used by the larger subunit of ATS ribosome. किस चीज के लिए? Sir, in order to bind with cistern. Those two glycoproteins are called as ribo, sorry for the inconvenience, riboforin first and riboforin second. Crucial information can be asked in the competition. Larger subunit uses which glycoproteins to bind itself with the cistern of here? Sir, two glycoproteins, riboforin first, riboforin second. A quick recap. What are cisternae? Sir, these are long, flat, tube-like, unbranched structures which show staking and are continuous with the outer membrane of nucleus. Their outer surface is studded with ribosomes, ATS, cytoplasmic ribosomes, by virtue of their larger subunit. Using two important glycoproteins, riboforin first and riboforin second, these two glycoproteins can be asked in your examination कि ये किस subunit of ribosome को किसकी surface के साथ जोड़ते? I hope every item is listening. Tubules. Let me show you the second structural component of these ER tubules. They are like this. Like this. Like this. They are like this. तो क्या दिख रहा है आपको ड्राइंग में? सर ट्यूब्यूल्स नंबर वन, और ऑल्सो ट्यूब लाइक स्ट्रक्चर्स, but they are branched, unlike sister. Unlike sister, these tubules are scattered in the cytoplasm. And what about sister? Sir, they are continuous with the outer membrane of what? Nucleus. But these sister are all together scattered, फैले हुए होते हैं cytoplasm. Third, their outer surface. Whose outer surface? Sir, sister sorry, tubules, is free from ribosomes. It is free from ribosomes. कौन से ribosomes? Sir, ATS ribosomes. माने unlike sister these tubes, which we call as tubules, the second structural components of ER, they are branched, they are not continuous with ER but are scattered, and unlike sister they do not have ribosomes on their surface, outer surface, I mean to say. I hope it's clear. Vesicles, vesicles, the third structural elements of ER, or third structural components of ER, like this, like this. तो क्या समझ में आ रहा है इस रेड कलर की ड्राइंग से अबाउट वेसिकल्स? नंबर वन, these are small oval or rounded ER elements. Number two, like tubules, they are also scattered in cytoplasm. Mane, vesicles are also not continuous with the outer membrane of what? Nucleus. Third, their outer surface is also free from ribosomes. माने आप लोगों को आइडिया मिल गया होगा, हमें कंपटीशन में पूछा जा सकता है सिमिलरिटी एंड डिसिमिलरिटी बिटवीन सिस्टर्ने, tubules and vesicles. I hope you people will be able to compare cistern, tubules, and vesicles with each other on the basis of their similarity and dissimilarity. Is that clear? Yes, sir, it is clear. I am assuming that you people are getting the things very clear. Now, if you remember, in the beginning, I had told you all the cell organelles which are included in endomembrane system. They have had one thing common. What is that common thing? They are single membrane bound. माने जो एंडोप्लाज्मिक रेटिकुलम के एलिमेंट्स होंगे, कौन से एलिमेंट सर? सिस्टर ने. कौन से एलिमेंट सर? ट्यूब्यूल्स. कौन से एलिमेंट सर? वेसिकल्स. There will be also membrane bound, they will be also having a single membrane around them. हाँ सर, होगा. एक बात. दूसरी बात, अभी मैंने थोड़ी देर पहले आपको कहा कि ये जो सिस्टर ने है, दे आर कंटिन्यूअस विथ आउटर मेम्ब्रेन ऑफ न्यूक्लियस. अभी मैंने आपको कहा कि ये जो ट्यूब्यूल्स हैं, दे आर स्कैटर्ड इन द साइटोप्लाज्म. सेम होल्ड्स ट्रू फॉर वेसिकल्स. माने जो ER एलिमेंट्स होंगे, ER एलिमेंट्स माने सिस्टर ने, ट्यूब्यूल्स एंड वेसिकल्स, they are scattered throughout the cytoplasm. Sorry, they are scattered throughout the cytoplasm. माने ये जो ER elements हैं, they are not localized in the cytoplasm. They are scattered. And this information you already. Now, if you allow me, I will show these scattered ER elements in the cytoplasm with your permission. Sir, permission granted. So let us consider. Consider what? Again, we have a cell over here. Suppose this blue color dibba represents a cell. Animal cell आप consider कर सकते, plant cell भी consider कर सकते हैं, आपकी मर्जी. Suppose this is a cell over here. हमें यहाँ पे दिखाना क्या है? सर आपको ER के एलिमेंट्स दिखाने हैं स्कैटर्ड, लोकलाइज्ड नहीं दिखा. इस सेल में साइटोप्लाज्म तो होगा. हाँ सर होगा ना. तो सपोज these yellow color dots are representing cytoplasm of the cell. Suppose these yellow dots are representing cytoplasm of the cell. मुझे यहाँ पे ड्राइंग दिखाने की जरूरत नहीं थी, बट उससे आपको फील आ जाएगा, अंडरस्टैंडिंग बेटर होगी, नथिंग मोर देन. साइटोप्लाज्म में हमें किन को दिखाना है? स्कैटर्ड ER को. इसीलिए सिर्फ ER और न्यूक्लियस दिखाएंगे, बाकी सेल ऑर्गेनेलीज होंगे ऑब्वियसली. ये है हमारा न्यूक्लियस. यहाँ पे दिस इज आवर न्यूक्लियस ओवर हियर. जिसके पास दो मेम्ब्रेन है, आउटर और इनर. कैन यू पीपल टेल मी विच कंपोनेंट ऑफ ER इज कंटिन्यूअस विद द आउटर मेम्ब्रेन ऑफ न्यूक्लियस? सर अभी बताया ना आपने, सिस्टर ने. तो सिस्टर ने दिखाते हैं पहले. दीज आर गोइंग टू बी सिस्टर ने. ये सिस्टर ने होंगी. सपोज दीज आर द सिस्टर ने. इनके आउटर सरफेस पे क्या होंगे? सर आप ही ने तो बताया, छोटे छोटे ग्रेन्यूल्स. उन छोटे छोटे ग्रेन्यूल्स को हम क्या कहते हैं? सर उनको हम कहते हैं राइबोसोम. तो ये मैं टैग कर रहा हूं इस चीज को एज सिस्टर. CISTERNAE. These are sister. Tubules are also continuous with the outer membrane of nucleus. No, sir, अभी आपने बताया था they are scattered. So let us show those scattered tubules in the cytoplasm. यहाँ पे थोड़ा दिखाएंगे ऐसे scattered, और यहां पे थोड़ा दिखाएंगे ऐसे स्कैटर्ड. और vesicles also continuous with the outer surface or outer membrane of nucleus. No, सर आपने बताया था वो scattered है. तो let us also show them scattered. like this. Mane, like these vesicles. The second structural component of ER, that is the tubules, they are also scattered. वही जो मैंने ऊपर लिखा, both tubules and vesicles are scattered in the cytoplasm. Overall, all the components of ER are scattered in the cytoplasm. They are not present along with each other. They are not localized. एक बात. दूसरी बात, सर आपने अभी कहा ER elements माने cisternate tubules and vesicles. They are single membrane bound, which means cisternate are single membrane bound, which means vesicles are single membrane bound, which means tubules are single membrane bound. Due to the presence of membrane around each structural component of ER, due to the presence of a single membrane around each component of ER, that is cisternate tubules and vesicles, we call them as, or this ER is famous as system of membranes. Which cell organelle is famous as system of membranes? Answer, endoplasmic reticulum. Why it is called system of membranes? Because its elements, number one, are scattered in the cytoplasm, and number two, they are all single membrane bound. Important information. अगर आपके एग्जाम में क्वेश्चन आ जाए, which of the following cell organelle is famous as system of membranes, what will be our answer? Sir, ER. Reason? Two. One, the scattered nature of its components. Second, all the elements of ER are single membrane. Now, as you know, each component of ER is membrane bound. तो जरा आप पहले ड्राइंग पे फोकस करिए. वेजाइकल का अपना मेम्ब्रेन होगा, हाँ सर होगा. इस मेम्ब्रेन का बाहर वाला साइड साइटोप्लाज्म और इनर वाला साइड हो गया ल्यूमेन, हाँ सर हो गया. ट्यूब्यूल का बाहर, ट्यूब्यूल के अराउंड जो मेम्ब्रेन होगा, मेम्ब्रेन के बाहर साइटोप्लाज्मिक साइड, मेम्ब्रेन के अंदर ल्यूमेन, हाँ सर सही है. सिस्टर ने सिंगल मेम्ब्रेन बाउंड, यस सर. तो सिस्टर ने के बाहर वाला साइड साइटोप्लाज्मिक साइड और सिस्टर ने के अंदर वाला साइड ल्यूमेन. वंस मोर, जब हम बात करते हैं एंडोप्लाज्मिक रेटिक्यूलम, इस एंडोप्लाज्मिक रेटिक्यूलम के तीनों कंपोनेंट्स के पास, किस किस कंपोनेंट के पास? सिस्टर ने, ट्यूब्यूल्स और वेजाइकल्स के पास क्या है सर? सिंगल मेम्ब्रेन. तो सिंगल मेम्ब्रेन का बाहर वाला साइड साइटोप्लाज्म का और उसके अंदर ल्यूमेन वेजाइकल. ट्यूब्यूल, मेम्ब्रेन आउटसाइड साइटोप्लाज्म, इनर साइड ल्यूमेन. सिस्टर ने का जो मेम्ब्रेन है उसका बाहर वाला साइड साइटोप्लाज्मिक साइड और उसका अंदर वाला साइड ल्यूमेन. I have shown that cytoplasmic site and lumen site in all the three components of ER with the help of yellow arrows. कहने का मतलब ये है कि ER की वजह से ये जो इंट्रासेल्यूलर मैट्रिक्स है, इंट्रासेल्यूलर मैट्रिक्स, आई मीन टू से साइटोप्लाज्म। कहने का मतलब है ईआर की वजह से ये जो इंट्रासेल्यूलर मैट्रिक्स है सेल का, दैट इज साइटोप्लाज्म, इट गेट्स डिवाइडेड into two compartments, one and second. Due to the scattered nature of ER, the cytoplasm gets divided into two compartments. One extra luminal compartment, and second luminal compartment. एक्स्ट्रा ल्यूमिनल कंपार्टमेंट ये है जहां पे मैं ऑरेंज कलर के एरोज लगा रहा हूं। एक्स्ट्रा ल्यूमिनल कंपार्टमेंट ये है, एक्स्ट्रा ल्यूमिनल कंपार्टमेंट ये है। माने सर आप एरो दिखा रहे हैं साइटोप्लाज्मिक वाले, साइटोप्लाज्मिक साइट रिप्रेजेंट्स द एक्स्ट्रा ल्यूमिनल कंपार्टमेंट। वेसिकल्स के, ट्यूब्यूल्स के और सिस्टरने के मेम्ब्रेन्स का जो आउटर साइड है, साइटोप्लाज्मिक साइड, दैट इज एक्स्ट्रा ल्यूमिनल कंपार्टमेंट। और ल्यूमिनल साइड माने मेम्ब्रेन के अंदर जो एरोज हैं। सर किस मेम्ब्रेन के अंदर? वेसिकल के अंदर। सर